It's my pleasure to be here speaking with you today. Thank you all for coming and participating in this exciting event. I feel like I'm following some very interesting talks. The bar has been set so high by my colleagues. This exciting research going on in the college, so it's really an honor to be here and be able to talk to you about what we do. So as was mentioned, I focus on cancer, and I'm an engineer. So this has not historically been the most common combination. I was originally trained as a mechanical engineer, and in fact, my PhD is in biomedical engineering. It had nothing also to do with cancer or disease. I worked on microgravity and NASA and bone density and things that I found very exciting. But like probably many of you in the audience, at some point, I became very angry at cancer. And so I decided to make a career switch, and I switched, and I moved into a cancer cell biology lab, and I did a 5-year postdoctoral fellowship on cancer biology, which was an interesting thing because biologists and engineers don't speak the same language. And so part of what you do when you make these interdisciplinary teams and you make these moves is you learn how to speak the other language. Now, I had good textbook knowledge of biochemistry. I thought I was going to do that before I went into mechanical engineering. I studied quite a bit of that. But going into a lab and being immersed like that was a totally different experience. It took years to learn the language of biology. But once you can be multilingual in multiple fields, you can create these interfaces in the discipline that are hard to find. 15 years ago, there wasn't very many people doing that. I tell people if I had had a meeting for engineering and oncology 15 years ago, I could have had it in my house. When I first started studying cancer as an engineer, many of my colleagues thought this was just a crazy idea. The physics of the tumor weren't that important. It just was what it is. You couldn't change it. And I would try to explain to them things like, well, certainly I can understand the physics of a bridge, but I can re-engineer it. I could make it collapse. And I'm going to talk to you a little bit about that microenvironment around the cancer cells. Can I re-engineer it? Can I make part of it collapse? Can I perturb it? Can I control it? And can I control it for the purpose of having a therapeutic benefit? And so that's the story I'm going to tell you guys about. And I'm going to tell you how ultimately it led from essentially seeing a problem on paper and thinking of it as an engineering problem instead of a biology problem, and how that led to what's now a drug in phase 3 clinical trials. But first, I want to set up the idea of how we think about cancer. So this is a very simple diagram of cancer, but what I want you to take home from it is these blue cells, cancerous cells, in this case carcinoma cells, but they don't exist alone. Turns out they have an enormous supporting cast that is essential for their function. So in a tumor, it's like any other organ in your body. It's just a chronically wounded, inflamed organ system that doesn't heal. But just like your kidney that we heard about, or your lungs, there's this epithelium, for instance, but there's a supporting cast. There's blood vessels. There's nerves. There's these workhorse cells that make all the structural proteins and scaffolds that hold that whole thing together. Right? There's an immune system. Cells going in and out, helping keep things clean, help get rid of infection, et cetera. Tumors are no different. The difference is they've co-opted many of these other cells and structures for their own benefit. They've co-opted parts of the immune system to tell the rest of your body, don't mind me, I'm fine, which is the great lie. Right? And so you want to get these engineered T cells into the tumor. They have to be able to surmount physical and chemical barriers to effectively infiltrate and treat a tumor. So we think about this tumor as a complex wounded organ system, and we think of it as a multi-scale problem. We think about studying cancer cells, but then how cancer cells interact with their neighbors. Maybe this, say, what we'd call a carcinoma-associated fibroblasts. These guys like to give a lot of chemical signals that keep the cancer cells happy and keep the immune system away. Right? And so we think about this connectivity, and then we think about the whole organ physiology. Turns out tumors are highly metabolic. They chew through resources. They have altered pH. It's a whole different landscape. So we start thinking about how these factors converge to drive disease progression. And the way we think of it is a little bit different. I'm sure many of you have turned on the news or a news story and heard about a new mutation, a gene. Or maybe some people have heard about themselves or family members that you harbor a gene that makes you more susceptible to cancer. Right? Cancer at its core is fundamentally a genetic disease. But how that genetic aberration manifests is dictated by the environment around the cells. So you can have one cancerous cell in one place do nothing. It sit there its entire life. It won't divide. It won't move. But then you put that same genetically aberrant cell in a different microenvironment. Now it may grow. It may leave that site and go to a new site. So this microenvironment around the cells influences how that genetic aberration is manifested and the consequences of it. And it turns out that in addition to biochemical signals, which we hear a lot about, just like I'm walking around on this stage, cancer cells have to use physical forces to move. So the laws of physics apply to cancer as well, right? Of course. And so how does the ground affect how a cancer cell moves? I'm on a hard stage. It's very easy for me to exert force and press and walk. Now, if I was on wet sand, I might be sinking in, or mud and kind of sludging through it, right? If I was on ice up here, which is not an uncommon feature of Minnesota, I'd be slipping around. I wouldn't be able to move that well. It's no different from a cancer cell. How that environment is physically, the physical properties of it dictate how that cell is able to move or not move. Likewise, there's patterns inside the tumor, and I'll tell you about this a little bit, architectures within the tumor that will dictate how efficiently a cell moves. For instance, if I want to go through that door right over there, the best line for me to do would be to run over all of you guys in some American Ninja Warrior fashion and get over there, right? Or, especially for you guys, would be more, I could take one of the paths that's already laid out, right? It turns out those paths of least resistance are no different. Those paths exist in tumors, and cancer cells follow them like highways within the tumor. So we can predict where they're going to go. But then we also redirect those highways or break them to stop the cancer cells from moving and progressing. But at the same time, leaving enough intact that the immune system can still take what's left and infiltrate. So that's what my lab does. One half of my lab roughly studies how these environmental cues impact the ability of a tumor cell to grow and ultimately metastasize, to set up a secondary tumor in another site. The other half of my lab thinks about how do we intervene therapeutically? How do we change the tumor landscape? Instead of just bringing in a drug, say, to target a specific pathway, we often do that in combination with more complex therapies, such as trying to re-engineer aspects of the microenvironment. And these can be chemical enhancements, and they can be physical. So we have funding, for instance, from the National Cancer Institute to work on specific ways to re-engineer the tumor microenvironment, and also from the American Cancer Society. And we also partner with local companies like Boston Scientific to think about local interventions we might be able to bring to bear. So this is an example of what I'm talking about. If you look at these white lines here, this is inside of a pancreatic adenocarcinoma. And these green cells are cancerous cells. And what they're doing is they're walking and migrating and moving along that network. These are the highways I mentioned where they walk and move and head out towards a blood vessel and ultimately metastasize. This all started 15 years ago when I was taking a microscope with live tumor cells and trying to look at how they moved. I just want to understand, how does a cancer cell move in a tumor? And I started to see regions where they moved and regions where they didn't, and regions where they could move at some great distance and regions where they couldn't. And so if I looked at the proteins around them, I started to see these patterns. And so it became an engineering problem about how do you quantify and correlate those patterns? How do you understand, is this pattern actually just a cool feature? It looks like a pretty picture on a microscope. Or are these structural elements driving the way the tumor cell moves? It turns out they are. And in fact, the features of this patterns, these alignment patterns for how cancer cells move, are predictive of patient outcome. And in fact, for breast cancer patients, they're more predictive than something like estrogen receptor or something you may hear frequently about on the news. So if these patterns are present, the likelihood of a poor outcome for that patient is much higher. So in patients like that, could you think about not just targeting the cancer cells, but in fact trying to take away these patterns within the tumor so that cancer cells can no longer effectively move? Something like this, where we've gone from these patterns, these are highways for which the tumor cells will move and infiltrate into local tissues. Can we bring to bear ideas that take away these patterns, take away these cues to slow and stop migration of these cancer cells? And so this is something that we actively work on in the lab, is not understanding how these cells move along these patterns to metastasize, but how do we destroy them? How do we perturb this? But something really interesting happened along the way. As I was doing this, we were moving into this space in 2009. We were thinking about how do we go from understanding how disease progresses to actually treating it. And I read this paper by a very prominent cancer biologist named David Tuvison. And I want to walk you through this slide for just a moment, because there's something as engineers that jumped out at me quite profoundly. Over here in green is chemotherapy. And you can see, unfortunately, in normal tissue, we get plenty of chemotherapy to it. So the healthy tissue gets bathed in chemotherapy just fine, unfortunately. And if you have tumors that you put in very permissive environments, in this case, you take away all those supporting cells. You have bundles of cancer cells, but very limited stromal cells to support them. You, again, get chemotherapy to those cancer cells quite effectively. Unfortunately, this green, or lack of green here, is what happens inside of a pancreas tumor. So pancreatic ductal adenocarcinomas are not effectively perfused with drugs. It turns out part of the reason why is because they actually don't have that many blood vessels. They're hypovascular. By that, instead of having an extreme amount of nutrition, growing new vessels, they actually have fewer vessels than a normal tissue, and so they exist in this extreme state of nutrient deprivation, and they thrive in it. But as a consequence of that, we don't have very good plumbing. So you might argue that a pancreas tumor is missing 75% of the plumbing you'd normally expect a building to have to bring fluid through it. But somehow it still thrives. And what's interesting is you see these little yellow dots. That's a red channel that is telling you where blood flows. So if blood flows through that vessel, it'll get a red signal. And if it has chemotherapy in it, it'll get a green signal. So it turns out all that signal is caught in these little blood vessels. So you have the drugs flowing through a fraction of those vessels. It's there, but then it never leaves the vessel and goes into the tumor. Right? It's trapped. And so the conclusion of this paper was, we need to grow more blood vessels. And as an engineer, I said, I don't think more blood vessels are going to do anything. Having another pipe running a drug through the building and out the other side isn't going to make a lot of difference unless you can get that drug out of the blood vessel and into the tumor. So we refocused part of our therapeutic approach and our stromal engineering approach to think about how do we change transport, molecular transport by diffusion and convection. How do we shift the way drugs would move in a tumor? And so we started this project to understand why drugs do not leave pancreas tumors. And this has turned out to be more important than ever because pancreas cancer is projected to double in about the next 10 to 20 years. If it was any other disease, we would call it an epidemic. But unfortunately, given its current survival statistics, as that number increases, the number of deaths associated with pancreas cancer will also increase. So after we sought to see why drugs don't move out, we realized that there's a sugar inside of pancreas tumors that when it hits water, it swells. And as it swells, it takes up space and it compresses the vessels around them. And it creates these very high pressures. So high that you get to about 100, 120 millimeters of mercury. Now, many of us, when we go to the doctor's office and think about our blood pressure, right, those are normal sort of blood pressure numbers. But that means if you took the best pump in your body and the pancreas tumor and plugged them in together, your heart would not have enough power to pump drug through the tumor because the pressure gradient in that tumor is so high. But by finding out what was causing those pressure gradients, we developed a strategy to destroy those pressure gradients, and that's this drug PEG-PH20. So I was at the Fred Hutchinson Cancer Research Institute, and then here working with a Dr. Sunil Hingorani, and from this work, we showed that if you ablate these pressure gradients, the vessels re-expand and drug flows back through the tumor. This drug is now in phase 3 clinical trial after very successful phase 2 data. And as the phase 2 data came out, there was one thing that was fairly miraculous. These are stage 4 patients with metastatic disease, so they have established tumors in their liver and or lungs. One of those patients had a complete response. We'll hope and pray that it holds. But one thing that's really clear is the proof of principle behind the idea of being able to see a fundamental problem in how cancer drugs are being administered and seeing it from an engineering perspective. Not necessarily as a biological problem, but as a first principle thermodynamics transport problem. And then thinking about ways that you might re-engineer the microenvironment. So don't target the cancer cells at all, target their friends. And then take all the friends away, take all their supporting casts away, and then just see how tough they are, right? It's like an old B movie where you had the little guy back there with the thing and the big bodyguards around him. And well, let's get rid of the bodyguards and see how this goes. It turns out the little guy was very scrappy, and he's still pretty tough. But we're getting pretty tough, too. These cell-based therapies that we heard about are doing a great job at starting to attack and fight these fights for us. So with that in mind, we're doing those kinds of things. Part of our efforts through the National Cancer Institute are to think about how do we re-engineer the environment, not only to get drugs there, but to tilt the balance, to suppress immune cells that are trying to protect the tumor while simultaneously allowing the body's resources to come and fight the tumor. And so we have a great team in our lab, and funding, again, from the National Institutes of Health, the College of Science Engineering, our Institute of Engineering and Medicine, local industry have all been very supportive of these efforts. And as we go forth, we're going to seek to try to move these things not just from understanding disease, but from translating them from our lab into the clinic so that we can try to combat this disease and save patients' lives. Thank you.